So I wanted to welcome everyone, and of course, good morning. On behalf of the American Association of Cancer Research, I want to thank everyone for joining us today at this wonderful workshop. I'm Liz Jaffe, and I'm the president-elect of AACR. I'm also the deputy director of the Sidney Kimmel Cancer Center at Johns Hopkins. The AACR is proud to be co-sponsoring this workshop for the third straight year with the U.S. Food and Drug Administration, and I've been thrilled to be a co-chair with Dr. Amy McKee, who's the Supervisory Associate Director within the Office of Hematology and Oncology Products in the Center for Drug Evaluation and Research at the FDA. As the treatment paradigm for oncology shifts from monotherapy to combination therapies, a more efficient process of dose selection in the early stages of study design will be of critical importance, and that's really why we're here today, to try to think about what we're going to do, how we're going to move from single-agent immuno-oncology drugs to combinations. So the goal for this year's workshop is to explore the unique approaches that can be used by all of us to move in this direction. Specifically in this workshop, we are focusing on best practices regarding patient and dose selection. We also need to focus on biomarkers for selection of those patients who are going to most benefit from combinations. And ideally, we'd like to begin to think about the novel endpoints that can define patient benefit. This workshop will have 3 sessions. The first session will be an immuno-oncology overview, looking at the scope of the problem in 4 different disease areas. The second session will explore key translational and design questions for immuno-oncology combinations. And finally, the third session will examine considerations to dose selection, optimization of IO combination products. One of the hallmarks of this workshop is the interdisciplinary forum with a wide range of perspectives from stakeholders throughout our immuno-oncology community, and that includes academia, government, FDA, industry. I'm happy to report that as of last night, we have had 673 people registered for this workshop, in addition to nearly 200 people registered to attend this workshop here in DC. We have almost 500 people watching this workshop via the live webcast. So thank you all out there on the website for watching us, and we hope you'll participate, and if you have questions, please send them to us. The AACR is continually looking for opportunities to engage on important regulatory science issues, such as dose finding for immuno-oncology combinations, and the AACR looks forward to having continued productive partnerships with the FDA. So I want to thank all of you for joining us today, and I will now turn this over to my fellow workshop organizer, Dr. Amy McKee, who will share an update on outcomes from our 2 highly successful prior oncology dose-finding workshops from 2015 and 2016. Thank you.
Good morning. I'm Amy McKee. So why are we here, and how have we gotten here? What is the FDA perspective on why this is our 3rd workshop on dose finding? So we've completed 2 dose finding workshops. 2 years ago, our workshop focused on only small molecules, if you can imagine. That's where cancer was at that point. And so much has changed since then. Our 2nd workshop focused on biologics, and this 3rd workshop is focusing on combinations of immuno-oncology agents, which could encompass all of the prior workshops plus this one. So the question is, what is FDA getting from this, and how have we possibly evolved from these workshops in our regulatory work? So what I'm going to do is give you 3 quick examples of what has changed in the past couple of years and how we've used our knowledge gained both at this and our expertise inside the agency. So the first example is ruxolitinib. This was a marketing application that was submitted to FDA for treatment of patients with non-small cell lung cancer with the EGFR T790M mutation, and it was discussed at an ODAC in April of 2016. So our clinical pharmacology team noted that ruxolitinib, the parent drug, was responsible for the efficacy. However, 2 metabolites were responsible for the safety issues of hyperglycemia and QTc prolongation. Through work with our pharmacometrics team in a population PK analysis, it was noted that dose exposure was flat across all of these doses. Furthermore, the ruxolitinib exposure efficacy relationship was also flat. So where did this take us? It meant that our clinical team pooled all the data from the various doses that were included in the application to get a reasonable estimate of the true effect of ruxolitinib on tumor response. So that's how it affected the efficacy evaluation. And how did it affect the safety evaluation? So for one metabolite with hyperglycemia, there was a very steep exposure safety relationship, which was also demonstrated with QTc prolongation and that metabolite. So the conclusions from this were that we had a flat dose exposure relationship based on pooled data from across different dose levels for safety and efficacy. We had an exposure efficacy relationship that was flat, and we had an exposure safety relationship that was extremely steep. And the conclusion from the clinical pharmacology team was that the dose proposed by the sponsor was not adequately supported in the application. So my second example, which has been discussed previously, is nivolumab, which received approval in 2014 for metastatic melanoma at a weight-based dose. This approval was based on analysis of ORR and duration of response for the nivolumab arm in a randomized trial. And the clinical pharmacology data included pharmacokinetic data from over 1,000 patients in the application. The exposure response relationship for efficacy was flat. The exposure response relationship for safety was flat. In September 2016, based on a population PK model, we approved a change from flat dose to solid tumor indications. And this was based on all the work that both the company and our team did internally to validate that there would be a small difference in exposure that would be unlikely to have a clinically meaningful impact on efficacy and safety in the solid tumor indications. And that is the dose that is labeled today. My third example is lenvatinib. This was approved in May of 2016 for renal cell carcinoma at an 18 milligram daily dose in combination with everolimus, and it was based on the efficacy demonstrated in a randomized trial versus everolimus alone with the progression-free survival and overall survival curves here that supported the application. The issue with this application from a dosing perspective was that almost all of the patients required a dose interruption or reduction. However, in light of the extremely game-changing efficacy, we approved this product with a PMR for dose optimization. The issue was, what is the dose that should be studied? And so our clinical pharmacology team and pharmacometric team used exposure response modeling to recommend a dose for the post-marketing requirement. Now, the modeling has also been presented at previous meetings, but the challenge in building this model for the pharmacometrics team was that the exposure was not constant over time with the frequent dose interruptions and reductions because that led to a biased exposure-response relationship. So they had to build a new model to recommend a dose for the post-marketing requirement. And the conclusion was that we recommended a lenvatinib 14 milligram dose with the option for up titration and to compare it to the lenvatinib approved dose of 18 milligrams in combination with everolimus with the primary objective to evaluate the incidence of intolerable grade 2 and grade 3 to 5 adverse reactions. So these are the three examples of how we have been evolving from a regulatory perspective and making regulatory decisions based on modeling and other methods within our clinical pharmacology and clinical divisions. You will hear more about some of these novel regulatory approaches to initial combination trials, in particular from one of our FDA reviewers, Tiffany Ricks, that have been submitted to the FDA. And our hope is to continue this relationship with all of our partners to further evolve how we evaluate these combinations, because there are so many combinations we could examine, about which we'll hear in the next session, that I'm not sure how we'll do it all in a reasonable amount of time for the patients who really need these products. So that is the end of what I was going to start with. And so as the moderator for the first panel, I'll just note that I'm going to introduce all the speakers at the start. If you have a burning question that cannot wait for the moderated panel discussion, we may have time for 1 or 2 questions at the end of each speaker, but if it can wait until the moderated panel discussion, there will be time for questions from the audience, both in person and online. But I will introduce all the speakers at the start, and then they will come and give their presentations. So our speakers for this session are Dr. Robert Vonderhind, Dr. David McDermott, Dr. Leisha Emmons, and Dr. Joaquin Belmont, and they're going to give us a perspective of what are the challenges for immuno-oncology combinations within their respective disease expertise. So thank you very much for coming, and I'll welcome Dr. von der Heide to the podium.
Good morning, everybody. Good morning to those listening in, all 614 of you. We're glad you're here. Thank you, Amy. Thank you very much, and Liz for the very kind invitation. And today, what I would like to discuss is the immuno-oncology perspective looking at pancreas cancer and what issues there are there that might lead us the way forward. So we're sitting in the middle of the cancer immunotherapy revolution. We're all aware. We've learned many things over the last few years, in particular the power of T lymphocytes to make tumors regress and potentially go away. And we've also learned that this is not a 100% winning activity and that there's a bittersweet quality to what we're doing. And we hope to do quite a bit more. Last week's FDA panel unanimous recommendation of the CART19 T-cell program is just yet another amazing development in this field, and yet we're mindful of how can we do better, and that's, I think, why we're here today. From my perspective within academics and at the University of Pennsylvania and the Abramson Cancer Center, and also discussing with many colleagues in industry, my impression is that this is the paradigm we're living in. With the exception of the CART19 experience, we're in the PD-1 plus X experience. So the future of cancer immunotherapy very often is PD-1 plus your pipeline, and that's it. And and there's an argument to be made why that's wise. I want to talk today about how perhaps, particularly in pancreas cancer, we need to switch that around, and I'll go through that. So pancreatic ductal adenocarcinoma is notorious, in fact, among tumors being resistant to the currently available immune therapies, in particular checkpoint blockade, where with the exception of MSI-high patients, there really are no reported responses. Pancreas cancer is now the third leading cause of death in this country. As of January of this year, more individuals will die of pancreas cancer than breast cancer. And that's the bad news. The good news is that combination chemotherapy is actually starting to have an impact, combinations such as GEM and nab-paclitaxel, which we'll talk about. As we thought about pancreas ductal adenocarcinoma and how can we use the immune system to attack, which I'm showing here, this desmoplastic stroma, along the way I met this person named Liz Jaffe, and literally over the last 10 years I've become her wingman as we try to put together some national efforts, along with many other folks, to take this problem on. So I do think that pancreas cancer is a window to understanding the immunology behind so-called cold tumors, those that have a scant effector T-cell infiltration in the tumor, which is the typical, not 100% situation in pancreas cancer. It's dominated actually in its leukocytic infiltration with myeloid cells, which we think are there because of the special oncogenic signaling processes of KRAS. We've noted now many others that there are a few missense mutations, not the indels that are seen in other tumors, and this doesn't create a high burden of neoepitopes as we understand it today. As I said, this tumor is resistant to the currently available immune therapies, and 3 recent studies, vaccine-only studies, randomized studies in this disease were negative, causing us to think a lot. But I should point out that in the setting of resected patients, those who go to surgery with the intent of cure, the higher the expression and the higher the CD8 infiltration into those tumors, the longer patients live. So the experiment of nature has been done. CD8 T cells can make a difference. We had the opportunity of looking at the data provided by the TCGA working group a few months ago and analyzed 134 primary pancreas cancers, these are humans, for the expression of a cytolytic signature which was comprised of granzyme A and perforin 1, shown on the left. You can see this S-shaped curve indicating that, in fact, most tumors show a low cytolytic index, but there are some in the orange box there that have a higher cytolytic index. And if one removes those 2 genes from the rest of the transcriptome and looks at by principal component analysis, you can see that cytolytic high tumors and cytolytic low tumors are completely different, even in pancreas cancer. We wanted to understand, is this on the basis of tumor intrinsic qualities? So we looked for the burden of neoepitopes and plotted on the bottom right cytolytic activity versus neoepitopes defined in a classical way. And you can see that there's not a correlation. In fact, it's a little bit inverse. Those patients with the highest cytolytic index are the ones with the lowest predicted neoepitopes for class 1 and vice versa. Looking further at TCGA transcriptome data, you can see in the upper panel the massive heterogeneity of primary pancreas cancers with regard to the expression of a number of immune checkpoints shown here and scaled in the heat map as indicated. Certain vulnerabilities highlight themselves, such as potentially VISTA, but I wanted to point out PD-L1 and how it's a rather lowly molecule in this disease, although it's a dynamic one. It's perhaps something to keep our eye on. You can see where pancreas cancer fits across the rest of TCGA. There are unique qualities of pancreas cancer when compared to other tumors, particularly melanoma and lung cancer. To understand whether T cells have a role in this disease and can be exploited, we turn to a genetically engineered mouse model of pancreas cancer, the KPC model, which is generated by introducing mutated KRAS and mutated P53 into the pancreas of these animals and watching by serial ultrasonography the growth of tumors in the pancreas as identified here. And that allowed us to observe these tumors growing in wild-type animals, but also compare it to animals in which we had pharmacologically depleted the CD8 and CD4 subset. And this data has been published, so I'll summarize the key points that we found. KPC tumors, like human pancreas cancer, exhibit a low mutational burden, an absence of predicted neoepitopes derived from these, a scant T cell infiltration, and resistance to checkpoint therapies such as PD-1 and CTLA-4. Spontaneous tumor progression in this model is identical whether or not there are T cells. So this is much different than the models upon which the immunoediting hypothesis was based, in which there was a huge difference in the phenotype whether or not T cells were there. Here, no. And T cells, furthermore, rising in T cell depleted mice grow unchecked, never rejected, in immune competent hosts, again, in contrast to the immunoediting hypothesis. So we introduced, hypothesizing that the problem was antigenic burden, we introduced the strongest antigen we could think of, which was full-length ova. And when we did that in this tumor model, it leads to the rejection of the tumor and T cell memory. And this doesn't happen in ova-tolerant animals, indicating the special quality of ova in this experiment was its neo quality. So is there a way forward? And as Liz and I have talked many times and with our colleagues, we speculate there is, that this is a glass half full example. Maybe in the absence of strong antigens, there actually is no Darwinian-like pressure from T cells in this tumor, and that the underlying pancreas tumor cells remain highly susceptible to T cells because they've never had to escape. And so there is a role, perhaps, of T cells if and only if we can provoke these T cells into the tumor microenvironment. So this is our paradigm, I think, currently in the cancer immunity cycle, in this now famous paper in Immunity in 2013. And in 2017, I think largely we're over here with most of the FDA-approved drugs over here, checkpoint blockade, where success is based on the pre-existing infiltration of T cells in these tumors. But if that's not the case, such as in most patients with pancreas cancer, I think we have to go to the left here and trigger a response. And an observation that we made in the KPC animal is that even in those ova-expressing tumors, if the animal does not have CD40 or does not have BATF3, so there's no dendritic cells, we don't get rejection. Priming is important, and it highlights the special role of CD40, which I'll come to. It also presents a different way of looking at things. Instead of PD-1 plus X, we need X, then PD-1. So that's actually my main point today. It may seem simple, but I think that's where we are. So with that logic in mind, Liz and I and so many other people have begun to think about combination immunotherapy for pancreas cancer, which goes far beyond single agent checkpoint therapy or even combination checkpoint therapy alone. We don't think this will work. We noticed this with great fanfare, FDA approved PD-1 for MSI-high defective mismatch repair tumors, including pancreas cancer. That actually has set our field on fire, by the way, because now we have something to look for, only 1% of cases. But it does also highlight that we can be successful even in this disease. What I'm showing here is not a complete list, but an exhaustive list of clinical trials which are open now, in which the idea is to vaccinate the patients and come in with a blockade of immunosuppressive features. And I think that in pancreas cancer, perhaps in other tumors, will be our way forward. Let me spend the last bit of time on the role of agonistic CD40 antibodies. We think this is critical and a special pathway for helping to prime T cells. The mechanisms are many-fold. 15, 20 years ago, it was predicted that the ability of CD40 to activate antigen-presenting cells and therefore prime T cells would be critical in tumor immunology. CD40 can also reactivate and educate macrophages and cause those macrophages to become tumoricidal. But in those experiments, we do not see long-lived immunological memory, which we think is ultimately the goal. These agents, mostly but not exclusively agonistic antibodies, not blocking, agonistic antibodies, have gone forward in the clinic. What has been observed so far, summarized very briefly, is that there is an acute activation of antigen-presenting cells that's possible even in tissues. There is a mild, moderate, transient cytokine release syndrome and elevation of liver function test abnormalities, which is entirely clinically manageable. And multiple CD40 agonists are now in clinical development, although most of them are first-in-human studies. Let me outline some myths that I have perceived over the last decade working on this pathway. It is stated, and I've seen discussed, that CD40 agonists will create an unmanageable and inedible severe cytokine release syndrome. That's not true. There is a dose dependency to this drug as well as others. We have seen liver function abnormalities, but nothing clinically significant. Not every CD40 antibody is built the same. Some require cross-linking, but not all. And that's important because this is not a single class. And the fact that some folks say there's no clinical activity, we actually have a fairly decent amount of clinical activity that we've reported on. In pancreas cancer, in combination with gemcitabine, we reported a few years ago a response rate, objective response rate of 24% with a median progression-free survival and overall survival that matches that of a gem and a Abraxane, with a metabolic response rate even higher, the majority of patients responding as evidenced by FDG PET. You can see an example of a patient from this study a few years ago treated with gemcitabine and CD40 and the reduction of the tumor. When we looked and inspected the tumor, hoping that we'd see T cells, we mostly saw macrophages, and it sent us back to the laboratory to understand how can we better induce T cell immunity. And here, we returned to the KPC model, and instead of just using GEM, long story short is we added nab-paclitaxel because the FDA had approved that for this drug. And it turned out that made all the difference. And what we were able to observe with GEM, nab-paclitaxel, and a prototype for agonistic anti-CD40 shown there is tumor regressions in the KPC model and long-term survivors that we had never seen before. And these regressions were entirely dependent on T cells and interferon gamma, as evidenced here, and we've published this recently. It depends on the nab-paclitaxel, interestingly enough, and it's the addition of nab-paclitaxel to GEM-CD40 that converted the mechanism to a T cell-dependent, macrophage-independent way. So having done that, we then identified what can checkpoint do. So if that's the X, can checkpoint blockade add something to this? This is data, again, from a recent paper showing that we can achieve long-term survivors when we combine GEM, nab-paclitaxel, CD40, and checkpoint blockade. This was considered kind of a crazy idea, but when you think about it, of the drugs I just said, of those 5 drugs, 4 of them are FDA approved, 2 for this indication. So we decided to keep going. Looking further in the mice, what we identified is that if this is the genetically engineered animal at baseline with scant T cell infiltration, following treatment with GEM, nab-paclitaxel, and the agonistic CD40, we're able to put T cells, prime T cells now, into the tumor microenvironment. And when those are activated with PD-1 and CTLA-4 or both, that's where we see the effect. This has now taken hold in the clinic. I'm showing you 2 clinical trials where CD40 monoclonal antibodies are used in pancreas cancer. I won't have time to go through them in detail, but to highlight to you the opportunities. So again, working with Liz and others at Stand Up to Cancer and the NCI-CITN and PanCan, there's an open clinical trial at a number of sites, including Penn, Hopkins, and University of Washington and others, in which patients with resectable pancreas cancer undergo treatment with chemotherapy and the Roche anti-CD40 agonistic antibody, both in the neoadjuvant setting and in the adjuvant setting. This is a 2-arm study, 10 patients per arm, and the primary endpoint here is safety and feasibility. A study that is now open and just appeared on clinicaltrials.gov is for patients, first-line patients who have metastatic pancreas cancer. This is a clinical trial shown on the bottom where the IND sponsor is the Parker Institute for Cancer Immunotherapy. I serve as the overall PI of this study. Each site in the institute has a site PI. And this is a clinical trial where patients will be treated in a phase 1b, phase 2 design and receive treatment on one of 3 arms: chemotherapy plus CD40, and here we're partnering with Apexigen and their agonistic CD40, chemotherapy and nivolumab, where we're partnering with BMS, or all 4. And the idea here is to look at 1-year overall survival and compare it to what we understand to be the case for gem nab-paclitaxel. There are special challenges in using CD40, and I would say any agonist. In oncology, we're pretty good at antagonists and understand the PK and PD effects there. Agonists are much more difficult. And so determining the optimal schedule of the use of CD40 has been a challenge. And also the sequence. For example, does it come before or after chemotherapy? And there's been quite a bit of interesting preclinical data in that regard. And so here we always use chemotherapy first, wait 2 days, and then give the CD40. Other ways of sequencing this are much less effective and can be highly toxic. We need to still understand the optimal route of administration. We're giving all these drugs intravenously, but that may not be the optimal way. And every CD40 antibody is a little bit different, and we're still yet to understand the importance and the priority of whether or not we need to use antibodies that require cross-linking to be activated. And of course, we're still searching for ideal partners, and as I said, the sequence. So I'll leave it there for today's presentation. I want to thank those in the laboratory, my laboratory at Penn, particularly those who've helped with the preclinical data. So many scientific and clinical collaborators, not only at Penn, but elsewhere, and acknowledgement to funding sources and the Parker Institute for making these studies possible. Thanks very much.
Good morning. My name is David McDermott. I'm from Beth Israel Deaconess in Boston and Dana-Farber Harvard Cancer Center. I'd like to talk to you today. I'd like to thank first the organizers for the invitation to speak on how we're improving kidney cancer therapy through novel clinical trial design. And what I hope to get across today is how what we're learning in kidney cancer might be applicable to other tumor types. Here are my disclosures. This isn't my talk, unfortunately. Can I give you the talk that I'd like to give? So I apologize for that. All right, so those are my disclosures. Okay, so while the application of VEGF and immune checkpoint blockade has improved outcomes for patients with metastatic kidney cancer, new strategies are clearly essential to overcome treatment resistance and improve patient selection. Preliminary preclinical studies have suggested that anti-VEGF might enhance the anti-tumor activity of immune checkpoint blockade by improving T-cell infiltration, upregulating MHC class I expression, and reversing myeloid immunosuppression. This work provided the rationale for combination clinical trials. We're now in an era where there are at least 5, by my count, very large phase 3 randomized trials with all very similar design for patients with untreated kidney cancer. Thousands of patients are going to be enrolled in these trials, comparing essentially VEGF plus immune checkpoint blockade versus the current standard of care, sunitinib. I think the question, though, for the field and so for all of us in kidney cancer is, are these approaches advances in immunotherapy? I think if I had to predict, I think many of these trials are going to be positive, and you're going to see FDA approvals for these combinations, but it's not entirely clear to me that we're improving immune therapy endpoints, and I'd like to focus on what I mean by that. So here's an example. A couple months ago, the FDA approved the combination of PD-1 blockade and chemotherapy for patients with lung cancer. And in my opinion, I think while there were clearly benefits to this approach, and this approach generated interesting hypotheses, it's not clear to me that this combination improves immunotherapy outcomes for patients with lung cancer. Why do I say that? Well, here's the design of the trial. This is the KEYNOTE-021 study, a study that patients were randomized to receive either carboplatin pemetrexed or chemotherapy plus pembrolizumab. The patients, as I said, had non-small cell lung cancer. They hadn't been treated before. It was 123 unselected patients. The primary endpoint was overall response. I think there were several aspects of this trial that I think are an issue. First is the trial design. In my opinion, I don't think this trial had the proper controls, and I don't think it was adequately sized. I think it was good for a hypothesis-generating trial, but not necessarily for a confirmatory trial. Here's an example of a hypothesis-generating trial that I think had some advantages in its study design. This is the EMOTION 150 trial. This is the first randomized frontline trial to explore anti-tumor activity of PD-L1 blockade with atezolizumab, both alone and in combination with the VEGF binding antibody bevacizumab versus the most commonly applied kidney cancer therapy at the moment, the VEGF TKI sunitinib. 305 patients with metastatic kidney cancer were enrolled in this hypothesis-generating trial, and the co-primary endpoints were progression-free survival in the intent-to-treat population and in patients with PD-L1 expression on greater than or equal to 1% of their tumor immune cells. The trial's exploratory endpoints identified for the first time in kidney cancer potential predictors of response and mechanisms of treatment resistance through the pre-specified analysis of gene expression signatures associated with the tumor microenvironment. Today, what I'd like to focus on is how the insights from this trial might educate the rational development of IO combination therapy in kidney cancer and beyond. So here's in contrast, in my opinion, I think the KEYNOTE-025 trial, 021 trial could have been improved if it had had a single-arm pembrolizumab alone, and we'll talk about why that is. It also could have been improved if it was larger. So once again, this is a good hypothesis-generating trial, but not large enough, in my opinion, to be confirmatory. Back to the EMOTION 150 trial. So what did we learn? Well, as presented at AACR earlier this year, while the Kaplan-Meier curves for progression-free survival in the ITT population were overlapping, in the PD-L1 positive population, curve separation was seen at the first treatment visit, that first scan that benefited the combination. This effect was also seen with atezolizumab monotherapy for the first time in patients with PD-L1 expression on greater than or equal to 5% of their tumor immune cells. This observation that the clinical activity of the combination improves as PD-L1 expression increases suggested that BEV might be improving the anti-tumor immune response. To explore this hypothesis further, whole transcriptome RNA sequencing data was used to classify patients' tumors into 3 distinct biologic subgroups representing tumor angiogenesis, pre-existing immunity, and immunosuppressive myeloid inflammation gene signatures. High and low gene expression was defined in relation to the median, which allowed us to categorize tumors as T effector high and low, angio high and low, etc. The heat map shows the relationship between the 3 previously published gene signatures, which were interrogated to evaluate the association with PFS and objective response in each treatment arm. Consistent with the prior PD-L1 analysis, the T effector high gene signature was associated with greater anti-tumor activity when the combination was compared to sunitinib, reinforcing our hypothesis about the potential impact of BEV in tumors with pre-existing immune responses. However, with longer follow-up, the PFS curves for the IO arm seem to come together, suggesting that the impact of VEGF blockade may be transient in some patients. The novel study design allowed us to explore mechanisms that might be driving the improved anti-tumor activity of the combination in the T effector high cohort. We went back to our transcriptome map and noted that the T effector high cohort could be divided into tumors with high and low expression of myeloid inflammatory genes. As several investigators have linked myeloid inflammatory cells to VEGF-mediated immunosuppression and poor outcomes in kidney cancer, we explored the impact that they might be having on response to therapy. So in the context of tumors with a pre-existing immune response, while atezo alone and the combination with BEV resulted in encouraging anti-tumor activity in the myeloid low subgroup shown here compared to sunitinib, atezo alone performed poorly in the group of tumors that were T effector high, myeloid high subgroup. The reduced clinical activity in this subgroup suggests a potential mechanism of innate resistance to atezolizumab monotherapy, which might be overcome by the addition of bevacizumab, possibly through its proposed ability to target myeloid inflammatory cells. So when we think about patient selection, I think what the Emotion 150 trial has suggested that maybe RNA sequencing will take us a step further than just PD-L1 expression with IHC, not only doing better at selection, but exploring potential mechanisms of resistance. So back to the carboplatin pemetrexed trial, the Keynote-021. I think one of the issues I also had with this trial is an unselected group of patients. I think the prior trial with pembrolizumab in untreated patients was a true advance for the field, where pembrolizumab versus chemo was shown to be better in patients with PD-L1 positive non-small cell lung cancer, a clear difference that allows selection in first-line patients. But what's the problem with an unselected subset of patients? Well, in my opinion, just going back to the kidney cancer data, if you look at nivolumab's activity in kidney cancer, over a third of patients' best response to PD-1 blockade in kidney cancer will be disease progression. My sense is in lung cancer, that number is probably at least as high, if not higher. So by approving a combination in an unselected group of patients, at least in a third of patients, you are giving them a treatment which is not likely to work and can only produce side effects. To me, that's not a step forward. Looking at how angiogenesis gene expression impacted response, I mean, I think this is the other, another reason why having a 3-arm trial is helpful, is you can look at the impact of your other agent in this combination. So looking at how sunitinib performed in tumors with angio-low or angio-high tumors, for the first time we're seeing a potential predictor of response to VEGF TKIs. We hadn't had one in kidney cancer before this. So in the patients who had angio-low tumors, sunitinib performing poorly, the IO therapies performing better, but in the angio-high subset, sunitinib outperforming the immune-based regimens, suggesting that in those tumors, maybe the patient should start out on a VEGF TKI. This trial design allowed us to figure that out. Now, obviously, all of this work is just hypothesis generating and needs to be validated in the ongoing phase 3 trials. But I think in this study, what we learned is that sunitinib probably primarily targets angiogenesis and is most effective in angiogenic tumors. Atezolizumab monotherapy has impressive activity in tumors with pre-existing or immunity, or the T effector high, myeloid low subset. Activity that in the long term is probably as good as the combination in these patients. However, the activity of the combination was good across many of these subsets. It seemed like the patients were deriving the most benefit of the combination in this T effector high, myeloid inflammation high subset, suggesting that maybe in the future this is the place we could use this combination to greatest effect. So also a word in the last few minutes about novel endpoints. In my mind, I think we should be focusing on endpoints that are immune oncology endpoints for our trials, not endpoints that we use for molecularly targeted agents or chemotherapy. Back to the 021 study, the primary endpoint of this study was overall response. I understand why that's important, and the trial clearly showed improvements in not just overall response and median progression-free survival, which led to its approval, but what about overall survival? When you look at the survival curves for this trial, they're overlapping. There's no difference in the number of deaths on these arms. And I would predict there's not likely to be. So in my mind, this is potentially an advance, but not a huge advance if we're not impacting overall survival. So when we think about combinations, a lot of them look good. A lot of them look good in small trials. But I think we need to focus on immuno-oncology outcomes if we truly want to make impacts on our patients' lives. What do I mean by that? Well, not every combination is going to be immune therapy. If you think about combinations, here's what we typically see with a lot of immunotherapies, a small number of patients benefiting, some of them benefiting after you stop the drug. And with VEGF blockade, by adding it, I would predict we're going to be moving the curve to the right. We're going to see higher response rates. We're going to see improvements in median progression-free survival. But unless we see this, I don't know that we've helped patients that much. We need to see tail of the curve benefits for the combinations. If we see them, then in my mind, these combinations will become standard of care in the front line. If not, then I personally would favor giving these drugs in sequence, VEGF, then PD-1 in some patients, PD-1 and then VEGF in other patients. And I want to believe that as we improve our selection criteria through novel biomarkers, this will help support sequential single agents for some patients. Because just remember, while we may show in the future that PD-1 and VEGF is better than VEGF alone, none of these trials are going to show whether PD-1 and VEGF in combination is better than the sequence. And for many patients, it won't be. What about other endpoints for trials? I mean, people have talked about landmark endpoints. I think those are interesting. Unfortunately, they take a while to develop. This is an endpoint we're trying to develop and bring to the clinic, is so-called treatment-free survival. We've seen these swimmer plots before in many publications with IO therapy, and it's these red intervals that I think are most important for our patients. These are our patients who benefit from treatment and then can stop treatment. They are off therapy in these endpoints, in these intervals. These are treatment-free intervals. Now, the concept of treatment-free intervals is not a new one to oncology. We see this in patients, for example, who have CLL, where they get treatment, get a benefit, and come off treatment. The difference, though, with immune oncology is when you stop the drug, the side effects don't always stop. So the way we look at a potential treatment-free survival would be free of both treatment for the cancer and significant treatment for the side effects. This might be a way of comparing regimens together if the goal is increasing the remission rate with these agents, focusing on what the patients truly want, which is these treatment-free intervals. Why is this also important? Well, we talked about side effects. Obviously, side effects get better when you stop the drug, but also the cost gets better. This is my sort of back-of-the-napkin calculations of the costs of combinations. One of the more prominent combinations now in the clinic is PD-1, CTLA-4. When you combine these, they're obviously more expensive. But if you're driving more remissions, the cost goes away. Not only that, the patients go back to their lives, which is what they want. So I think we need to be focusing on these treatment-free intervals and judging combinations in the future by whether they produce more of them. So I think in conclusion, I think the rational application of IO therapy in kidney cancer and beyond is going to require some collaborations, some discussions with the FDA and the NCI about innovative trial designs and permission to use some of these novel endpoints that I talked about. We also may need to get HMOs involved. I mean, I think one of the ways to speed development would be to get places like Cigna and Kaiser to fund our studies, because then we can look at stopping treatment and looking at these benefits off treatment. And we need to focus on the goals. So what are our goals? We want to improve patient selection. I think the Emotion 150 trial gives us a sense that next generation sequencing may do that. We obviously have to confirm that work. We obviously have to do a much better job of customizing the appropriate duration of treatment. You're going to hear a lot more about that during this conference. And finally, increasing the remission rate so there are more patients living off drug. If we do that, then you can change the name of the organization to the Food and No Drug Administration. Thank you for your attention. I apologize for the screw up in the beginning.
Good morning. I'd like to thank the organizers for the invitation to come and speak to y'all today about breast cancer immunotherapy. And the title of the talk is Provoking Breast Cancer Immunity with Strategic Immunotherapy Combinations. These are my disclosures. So one of the challenges with developing immunotherapy for breast cancer is that historically it's really been considered a non-immunogenic tumor. And this slide just shows you a stain of an untreated breast tumor for CD3 positive T cells. And you can see that some breast cancers on the left are cold and they really have no T cells in them. But there's a few that actually have a fairly robust T cell infiltration. The cold tumors tend to be more likely to occur in breast cancers that are ER positive. The breast cancers that are triple negative, that don't express the estrogen receptor, the progesterone receptor, or HER2, tend to be more likely to be inflamed. And the HER2 positive breast cancers also are more likely to be inflamed than the ER positive breast cancers, but somewhat less likely perhaps than the triple negative breast cancers. It's clear now from a lot of data sets that tumors with poor prognostic factors, those that are ER negative, PR negative, of high grade or lymph node positive, tend to have higher T cell infiltrates at diagnosis, higher numbers of CD8 positive TILs, and a higher T cell to FOXP3 Treg ratio. Predicts better clinical outcomes in terms of complete pathologic response to neoadjuvant chemotherapy, disease-free survival, and overall survival, except for in ER-positive breast cancers. Importantly, both triple-negative and HER2-positive breast cancers are really high-value targets for cancer immunotherapy. Triple-negative breast cancer because it tends to be inflamed and there are no approved targeted therapies for that disease, leaving us only with chemotherapy. And HER2-positive breast cancer because we have a lot of drugs for that disease, and some of them are potentially synergistic with immunotherapy. ER-positive breast cancers present the challenge of transforming tumors from cold to hot for breast cancer patients. So from a clinical point of view, we classify breast cancers for standard treatment by whether they express the estrogen receptor or progesterone receptor and will respond to endocrine therapy, HER2 and respond to HER2-directed therapy, or none of those, which leaves us with chemotherapy. So that's a pretty simple classification scheme. If you look genomically, you can classify breast cancers into an array of different types. These are the major ones shown here on the left. The HER2 positive, the basal-like, which is triple negative, luminal B and luminal A are ER positive. But you can see that these aren't clean distinctions. So if you look on the right-hand side, on the top are triple-negative breast cancers, and you can see that within these that are classified as triple-negative genomically, some of them will express the ER and some of them will express HER2. And the same is true for HER2-enriched, that's a heterogeneous group, as well as with the ER positive. So the lines in these classification schemes really are blurred. The same holds true for the immune features of these cancers. So if you look at luminal A and luminal B, they are much less likely to be lymphocyte predominant at only about 3%. They have fewer stromal and intratumoral TILs than either HER2 positive or triple negative breast cancers. And again, TILs at diagnosis are not predictive for those tumors. So very different immune phenotypes and immunobiologies in those tumors. HER2 enriched are more likely than ER positives to be lymphocyte predominant, about 11%. A similar rate for that in triple negatives. Both HER2 positive and triple negative breast cancers are more likely to contain TILs at 15 and 20% in the stroma, as well as intratumoral at 3 and 5%. And again, TILs present at diagnosis are predictive of response and clinical benefit. So there are several data sets that have been reported for targeting the PD-1 pathway in breast cancer, and this is the first large data set reported, and this is for the anti-PD-L1 agent avelumab. This is a trial that looked at 168 patients that were unselected for either subtype or PD-L1 expression, and you see the spider plot on the left. The objective response rate was only about 4.8% in this unselected patient population. However, there were some patients that did benefit, and we need to develop ways to really pick these patients out. There was 1 complete response and 7 partial responses. This is data for the anti-PD-1 agent pembrolizumab, and this was in PD-L1 selected patients. You can see the activity of this agent on the left in triple-negative breast cancer with a response rate of 18.5%. Again, a complete response as well as 4 partial responses, and many patients with prolonged benefit from treatment. On the right, you can see the activity of this agent in ER-positive breast cancer. Again, this was PD-L1 selected. Of note, they had to screen a lot of tumors in order to identify this small group of patients for testing this agent, and the response rate was somewhat lower than for triple-negative breast cancer at about 12%. Here, there were no complete responses, and there were 3 partial responses. And in breast cancer, you do see evidence of pseudoprogression with these agents. This is the data for the anti-PD-L1 agent atezolizumab in PD-L1 selected triple-negative breast cancer. Peter Schmidt presented this this past year at AACR. You can see the spider plot on the left with many patients progressing, and if you take those away, you see that there is a subset of patients who derive significant response. The median duration of response is 21 months by both RECIST and immune-related RECIST. And you can see that, again, that you see this evidence of pseudoprogression in that there's clinical benefit in some patients who actually are classified as progressive disease by standard RECIST. These are the response rates. So overall, in 112 patients, the response rate was 10%. If you look at patients who expressed PD-L1, the response rate rose to about 13%. And importantly, if you look in patients who were treated first line, the response rate appears to be higher at about 26%. Again, small numbers of patients here, only 19, but this is hypothesis generating and suggests that treating these patients earlier with immunotherapy is likely to be more beneficial. This looks at overall response or overall survival by response status. For the patient group overall, the median overall survival was 9.3 months in all patients, with landmarks at 1 year of 41% and 3 years of 22%. Importantly, the people that had a complete or partial response had 100% survival out to 2 years. This data continues to mature. And even the people that had stable disease had significant survival benefit, as you can see here. This data is by standard RECIST. You can see this tail of the curve here for people with a progressive disease. This really reflects the pseudoprogression, which when you look by immune RECIST, this tail goes away. So important to really thoughtfully assess the response of these patients to these agents so you don't take someone off therapy who's likely to benefit. This begins to drill down into patient selection and biomarkers that can predict response. So if you look at overall survival by PD-L1 status, you can see that the survival at 1 year for PD-L1 positive versus PD-L1 negative patients was 45% versus 37%, so this was somewhat predictive. Again, you have this tail of the curve out to 3 years for PD-L1 positive patients with an overall survival of about 28%. If you look at the association of response and survival with TILs, there was a significant association of both. You can see that the response rate is higher in patients with high levels of TILs, as shown here. And importantly, the overall survival was much better in patients with greater than the median level of TILs of greater than 10%, relative to those with less, with a p-value of 0.0028, and this is by multivariate analysis. Here you can see that in TIL low tumors, the median survival was 6.6 months, and TIL high was 12.6 months. So this data actually is very similar to the data more recently reported at ASCO by Sylvia Adams for pembrolizumab in metastatic triple-negative breast cancer. This is a cohort of 170 patients who were enrolled and treated. 105 or 62% approximately were PD-L1 positive, and about 64% were PD-L1 negative. You can see the response rates are fairly low across all of the patient cohorts, about 4.7%. This reflects the avelumab data somewhat. In a different cohort of patients who were treated first line and were PD-L1 selected, the response rate was 23%, very much like the first line data for atezolizumab, again suggesting that selecting for PD-L1 and treating earlier selects for a patient population more likely to respond. So PD-L1 appears to be an imperfect biomarker. It can enrich for responses. TILs appear also to be predictive of benefit, and the context really is important in terms of how these patients do. So there's two primary challenges for the field now. One is deepening responses to single-agent immunotherapy. The second is converting non-responders to responders, because the fact of the matter is the vast majority of patients with breast cancer don't respond to PD-1 and PD-L1 blockade. This is a slide that just summarizes some selected promising combination immunotherapies for breast cancer, and it gets back to Bob Vonderheid's comment at the beginning that we're really kind of in an era where we're thinking about PD-1 and PD-L1 plus X. So this illustrates PD-1 and PD-L1 as a backbone here, and then building on that by adding a variety of different agents with significant rationale actually for the combination, which puts us in the position of really having to try to prioritize which ones are the most important to take forward. In thinking about these combinations, the important variables are the drug, which chemotherapy agents are the best to combine, the dose of the drug, the sequence. We just heard from Dr. McDermott about sequencing issues. And what this cartoon doesn't show is the issue of individualization to the phenotype of the tumor and the genotype of the tumor. So considering mutated versus shared antigen load and different mechanisms of immunoregulation present in the tumor, which will really drive which other immune checkpoint inhibitors or immune checkpoint agonists to combine with targeted therapies. So I'll just talk a little bit about the four that I've highlighted with an arrow. This is data that shows the combination of PD-1 and PD-L1 blockade with standard chemotherapy in triple-negative breast cancer. These are patients with metastatic disease that are not PD-L1 selected. And this data looks at the combination of atezolizumab with nab-paclitaxel. Overall, in this group, the response rate was 41.7%, but consistent with the idea that first line may be better. In the 9 patients treated first line, the response rate was actually 67%. Relative to patients treated second line or greater, where it was 25 to 28%. So taxanes actually have immune modulatory activity. They promote antigen release. They can signal through the toll-like receptor 4 and augment dendritic cell activity and antigen presentation. So good rationale for that combination. Nab-paclitaxel also doesn't require steroid pretreatment. Another study has combined pembrolizumab with eribulin. The response rate here was 33.3%. And again, eribulin has immune modulatory activity. In addition to antigen release, it decreases immune suppressive cells within the tumor microenvironment. So this I just included to stimulate some thinking about novel trial designs. This is a trial that I did in collaboration with Liz Jaffe based on some preclinical modeling that she'd done in her lab. And it looked at a fixed dose of vaccine in combination with a variety of low doses of chemotherapy, cyclophosphamide given 1 day prior to the vaccine to mitigate regulatory T cells, and doxorubicin given 1 week later to promote T cell expansion. We tested different doses of cyclophosphamide in a 3x3 factorial design, and then ultimately mapped it out by mapping the induced HER2-specific antibody response on this 3x3 factorial matrix, and determined that the optimal doses of Cy and Dox to include with the vaccine were 200 mg per meter squared of Cy and 35 mg per meter squared of Dox. So the advantage of this type of design is that you can test the optimal combination of multiple interacting variables using as few resources, i.e. patients, as possible. And you can really adapt this is brainstorming a little bit, to think about different variables. So possible inputs might be a dose-by-dose question. Another option might be a dose on the y-axis by schedule question. And different possible outputs here for this analysis would be components of the immune response, now that we have more sophisticated ways of looking at that. clinical response if you're lucky enough to have a clinically active intervention, as well as potentially toxicity, look at toxicity in the context of dose. So there's a lot of promise in terms of combining immunotherapy with HER2-directed therapy, both immune checkpoint blockade, where you can see in HER2-positive tumors that are untreated, they grow out fairly quickly. If you treat them with either HER2-directed therapy or PD-1 alone, you get some benefit, but the two together result in the best. This looks at the combination of anti-PD-1, CTLA-4, and Kadcyla, and you can see that you get the best tumor-free survival with a combination of all three agents. Herceptin-based therapies actually have a lot of immune modulating activity, both promoting antibody-dependent cellular cytotoxicity and actually priming the immune response. And Kadcyla, which is Herceptin with a chemotherapeutic agent attached to it, the chemotherapeutic component of it actually can activate dendritic cells and promote immune priming as well. This looks at the combination of HER2-directed therapy in combination with vaccination in a tolerant HER2 model where the vaccine alone doesn't work. With cyclophosphamide to get rid of Tregs, you get some benefit. With a HER2-directed antibody, you get more benefit, and you get the best benefit with all three together. Triple therapy resulted in the greatest T-cell response, and this translated in the clinic pretty well, where a vaccine alone gave a very modest T-cell response. With chemotherapy, it was slightly higher, and it was the best with the addition of Herceptin. This is a cross-trial comparison here. And then finally, what about those cold tumors? There's another type of vaccine strategy with intratumoral vaccination. This looks at activating the STING pathway. T-cell inflamed tumors in humans typically have an interferon beta transcriptional signature, and STING is the critical receptor to activate immune cells, including dendritic cells. Tumor-derived DNA can induce interferon beta by tumor-resident DCs through this STING signaling, so you can have cyclic dinucleotides that therapeutically activate this pathway, induce interferon beta, and activate intratumoral tumor-specific CD8-positive T-cell priming. This looks at the evaluation of this approach in a paired mouse model system of non-tolerant mice, the FABN mice, and the tolerant HER2 transgenic mice. The non-tolerant mice have inflamed tumors with lots of T cells, both CD4 and CD8, whereas the non-tolerant mice really have a paucity of T cells, both CD4 and CD8. If you look at the intratumoral injection of this agent into tumors in the FABN mice, which are not tolerant, you can see that you get a good tumor rejection and actually protection from a second challenge just with the intratumoral injection of this agent alone. In contrast, in the tolerant mice, you get a little bit of delay, but by no means the best type of an effect. If you isolate the T cells that are present in the new transgenic mice and look at the immune checkpoints expressed on them, you can see that for CD8 positive T cells, you have lots of PD-1 in the tumor microenvironment relative to the spleen, as well as lots of OX40 in the tumor microenvironment relative to the spleen. And testing that combination preclinically, you can see that the triple therapy results in the best tumor-free survival relative to intratumoral injection alone or checkpoint blockade alone, and you actually get the greatest infiltrative T cells as well as the greatest activation of antigen-specific T cells here as well. And then I just threw this slide in. We're talking a lot about the tumor microenvironment and drugs to affect this part of the tumor biology and the immunobiology of the tumor. There are many things that are superimposed on this, including treatment history, the obesity that may be present in the patient, the BMI, the microbiome, and other hormonal factors that can come into play as well. So it's important to keep those factors that can influence response to immunotherapy in mind as well. So what I've tried to show you is that breast cancer can be immunogenic, contrary to historical beliefs, but most breast cancers inherently are not, so we have to figure out a way to transform them. Multiple layers of regulation within the tumor microenvironment shut down the immune response. Vaccines and STING agonists can prime the tumor microenvironment for response to immune checkpoint modulation, whether it be blockade or agonists. Standard cancer therapies can augment the activity of immunotherapies. The future is really in combinations, which should have synergistic clinical activity, ideally, but may come at a toxicity cost that we have to keep in mind. And we need to do smart trials that elucidate mechanisms of response and resistance in patients. These are my acknowledgments. And I really want to thank the patients who participate in all these trials, because without them, we couldn't advance to better treatments so that they can live longer, happier lives. Thank you.
Hi, good morning, everybody. My task will be to discuss about strategic immunotherapy combinations and sequencing in urothelial cancer. And also I will go a bit through the doses. As has been mentioned, initially with IOs, we haven't seen any dose limiting in several trials, so it's quite difficult to select the dose. And we know that the exposure response toxicity has quite a flat dose, as has been mentioned in the beginning of the presentation. And with combination, probably this is going to change, and we might need to tune a bit our trial design. So those are my disclosures. We are in exciting times for urothelial. After 20 years now, we have agents that provide some prolonged responses and survival benefit. But we know that only 15–20% of patients with bladder cancer are benefiting from immunotherapy single agent. And the future is obviously going to be combination or sequential use of these new agents. My lecture will be divided in three parts. First, I will review what's going on in terms of new combinations of IO or IO-targeted agents. The second, I will discuss a bit about the trial design and settings that we can take the advantage to optimize our trial design. And in the end, I'm going to talk about the new findings that have been recently presented that probably are going to help us to select patients in order to derive a higher benefit when giving combination immunotherapies. So potential synergistic combos is the first part. We have standard CTLA-4, PD-1, PD-L1 combinations, IOs with other immune approaches. And the first question that comes to our mind is combination better than sequential? And you have seen this Checkmate 67, although this was not a trial design to analyze combination versus sequential, because patients, the sequential therapy was not defined. But we can see that there is no that much difference, and these are at the cause of a high toxicity, so 58% versus 20%. And we can see that nivolumab versus ipi/nivo is not that much different. In bladder, although we do not have randomized trials comparing combination versus single agent, we have the results of this Checkmate 32 that combine ipi/nivo in patients previously treated. And you can see here the survival is 10.2 months. When we compare that to although as mentioned, this is not fair because I'm comparing phase 2 with the phase 3. When you see the survival obtained with the Keynote 45, the same survival is being observed. So obviously, we need randomized trials to address this question if combination is superior to single agent. In terms of combinations, we have now several trials ongoing in first-line therapy in bladder, exploring combination of IOs. This is the DANIF trial. This trial has close to patient accrual, so we are going to answer the question if combining durvalumab, tremelimumab, those are PD-L1 plus CTLA-4, is superior to monotherapy or superior to standard of care chemotherapy. And this is without patient selection. This is the all-comers, patients being fit or unfit. And there is another one that is accruing, comparing nivolumab in combination with ipilimumab compared to standard of care. So these trials in first line will provide some answers if a combination is superior. What about the new IOs? We know that there is an oral IDO inhibitor. I'm presenting this data here because some preliminary results have been presented at ASCO this year. So IDO is an interferon-induced intracellular enzyme that catalyzes the first and rate-limiting step of tryptophan degradation, and these lead to the immunosuppression. So if we combine this IDO inhibitor with pembrolizumab, like this trial. This is the trial that was presented, the Keynote 37. And you can see here in terms of the way that the dose was found to move to this expanded cohort in bladder. Initially, the recommended dose of pembrolizumab was 2 milligrams per kilogram, because this is coming from the very beginning, with different doses of epacadostat, that this is oral IDO agent. And then you see here that we jump to this pembro 200 milligrams, because this is the approved dose. And I want to remind remind you this JNCI paper published recently by Goldstein showing that probably we can save money if we use this dose that is a more personalized dose. So anyhow, and obviously we need to wait for the paper. We don't know, but finally the dose that was selected was this of epacadostat of 100 milligram with 200. And the results of this combination are quite nice, although it's, as mentioned, it is a phase 2 study. You can see that 40 patients with urothelial carcinoma, the overall response rate, 35%. This is more than this 20% that we usually see with pembrolizumab alone. And you can see here in patients having PD-L1 overexpression, the response rate goes up to 64%. Presumably, although, as mentioned, it's a phase 2, maybe patient selection, these might be potentially an attractive way to treat bladder cancer patients, especially in those having the PD-L1 expression. Another agent that is being tested in bladder cancer is, again, an antibody that is an agonist. And this is a 4-1BB. This is against CD137. It seems that it is able to alleviate T-cell exhaustion post-IO, induce CD8 T-cell activation, CD4 T-cells, and also natural killer cells. There is a phase 1 trial combining this agent named utomilumab, plus pembrolizumab, where you can see here no dose-limiting toxicity, again, is seen in the dose range used with this agent. So it's very difficult to select a dose for further development. So this combination is, well, here you can see the responses seen in different tumor types, renal cell, non-small cell. So this is an active combination. This was a phase 1, heavily pretreated patients, but at least the combination was tolerable. And now this is moving to different tumor types. You can see here the design of this trial. Obviously, avelumab single dose is the one that is approved of accepted as single agent, but for utomilumab, the dose optimization will follow with a standard expanded cohorts, and then if there is no dose-limiting toxicity, this will move to expanding in different cohorts, including bladder cancer. So obviously, this is, I believe, this is a nice way to design a trial, and as mentioned, this is going to move forward pretty soon. Another IO is the OX40. This is different from the CD40. OX40 is a CD134, not to get confused with Dr. Van der Heijden that presented before. And this is, again, an agonist monoclonal antibody that in the presence of the T-cell receptor stimulation and produce an enhanced T-cell response, and also can diminish the inhibitory effect of regulatory T-cells. So there are trials, including bladder cohorts, but as mentioned, there is no patient selection. This is for all comers, just trying to synergize, combining two of these new IOs. So this is the trial, OX40 alone or in combination with these other agonists that 4-1BB. So this is, as mentioned, trying to synergize. There are other trials ongoing with other IOS agents, anti-CSF1, and some responses, interesting responses have been seen in bladder. We have combinations with the TIGIT. And then a new ways of combining the IOs is with like antibody-drug conjugates, and also an interesting approach. This is being done at the Farber with different patient cohorts. You can see bladder, RCC, melanomas, where we are combining nivolumab with a personalized vaccine using neoantigens, using personalized peptides. So probably this is a way, and this has been also already presented during the breast lecture, is a way to more targeting, to target more in a more precise way the tumor antigens and optimizing with a standard checkpoint inhibitors. And then we have other potential combinations, that is the IOs with targeted therapies, and these might be more personalized for patients. One of those is the combination of an IO with cabozantinib. This is a work done by Andrea Polo, where she analyzed the immunoregulatory effects of cabozantinib that is approved for second-line renal cell carcinoma. And it was observed, as shown here, a decrease in Tregs among CD40 cells, an increase in PD-1 expression in the Tregs, and a decrease in CTLA4 expression also in Tregs, presumably producing an immunomodulatory property that could be used to combine with a PD-1 inhibitor. And here's the trial design. So this trial was presented preliminarily at ESMO. More data has been presented with a combination of cabozantinib, nivolumab, and ipilimumab. There is no patient selection. This is like a dose finding looking for safety. Sorry, how can I go back? Here. Okay. So those are the responses seen in different tumor types, including GU tumors, bladder, non-traditional histologies like squamous cell carcinoma of the bladder, penile, and interesting responses, overall response rate with a combination of cabozantinib and nivolumab of 43% in a heavily pretreated patient population. And this has shown to be quite tolerable. And then focusing more on trying to target different genomic profilings of bladder cancer, we know that like breast cancer, now based on RNA sequencing, we have the luminal type and the basal type. And what we have seen with atezolizumab, that it is the cluster 2, the luminal 2, that is able to respond more frequently to atezolizumab. And because we know that, for example, the cluster 1 is the one that is enriched for FGFR3 mutations, and those are patients that the likelihood of responding is pretty low, why not to combine FGFR3 inhibitors with immunotherapy? So, and this is the type of trial design that we started at the Farber. So combining pembrolizumab with B701, it might be we are targeting these 2 different RNA subtypes that can be found in bladder cancer. So unfortunately, this trial was activated in 2016, and because of some concerns of potential toxicity, is now on hold. But at least this is an attractive way to expand the way that immunotherapy treat bladder cancer. Let's move to the second topic, that is the taking advantage on the strategic designs and specific settings. And one important strategic area in bladder is the neoadjuvant setting, because with that we can get the biopsies, we can treat the patient, and then we have the cystectomy, and we can do correlation on what's happening with the treatment. There are several trials combining mainly chemotherapy, because chemotherapy is a standard of care in the neoadjuvant setting. And there are trials in patients who are unable to receive platinum, meaning that you can use combination of two IOs in this setting. I bring this here, this design, because this is coming from the breast world, and probably this needs to be applied to bladder cancer, is what is called the adaptively randomized neoadjuvant bladder trial. And here, and as mentioned, this is coming from breast cancer trial. The SPY2 is a biomarker-rich clinical trial with multiple platforms and serial tumor specimens. So a lot of studies are being made to identify the most likely to benefit based on a statistical design, meaning that this is a way that we can screen combinations pretty quickly and being able to decide what's the best combination for a specific group of patients. Another way in advanced setting is obviously the match type of trial design, and we have examples of that. There is a trial now accruing patients, and based on the profile, durvalumab, that is a PD-L1 inhibitor, is being combined with different AstraZeneca compounds. The one targeting the FGFR 1, 2, or 3, this is a tyrosine kinase inhibitor, different from this other compound that I explained before that was a monoclonal antibody. Patients having mismatch repair or DNA alterations, and we're going to see that this is an interesting area, newly discovered for bladder. Combination of durvalumab plus olaparib might be a good option for these patients. And obviously, monotherapy is here as a single arm, and the WEE1 inhibitor is also included in one of these arms. So genomically driven trial assigning combinations based on the specific genomic alterations. And then finally is concomitant versus sequential, and here I want to focus on chemotherapy. So chemotherapy, we have been using chemotherapy forever in our patients, and when combining with chemotherapy, we need to put the chemotherapy in the right context. It's not like to put it together and see what's going on. Initially, companies were very reluctant to combine chemotherapy with immunotherapy, presumably because you are killing the lymphocytes giving chemotherapy. Subsequently, we have seen that this is not happening, and the responses are seen in a synergistic way. So in bladder cancer, the first example was this trial combining gemcitabine cisplatin plus ipilimumab in patients with metastatic urothelial carcinoma, and this was a completely negative trial. No, although this was a single-arm phase 2 trial, no increased responses were seen combining EP plus gemcitabine and cisplatin. And then, as mentioned, there are first-line trials trying to answer this question. As mentioned, there are 4 first-line trials two combining IOs, and these others combining chemotherapy and immunotherapy. This is the keynote, putting together pembrolizumab plus platinum gemcitabine versus chemotherapy, or this other one that is combining gemcitabine carboplatin plus atezolizumab. There are trials in Europe designed just for logistic reasons. Bimflunin is approved in Europe, and why not to still try to have bimflunin in place? And there is this trial design. This is an IST trial where bimflunin pembrolizumab is going to be compared to bimflunin without no rationale, just because of the regulatory approval of bimflunin in Europe. So what about the best way to combine chemotherapy and immunotherapy? And this is an interesting paper, and mainly this reference where the different actions or ways to combine chemo and immunotherapy are addressed. It's an issue of the type of drug, the dose, and the schedule that might matter when combining chemotherapy and immunotherapy. And one of the things that have not been addressed is giving in an adequate way chemotherapy with immunotherapy. Based on this paper, we have designed this trial that is in patients who are unfit, first line. And in this case, you can see that this is a randomization to a standard chemotherapy for unfit patients, carboplatin gemcitabine, versus avelumab plus carboplatin gemcitabine, but given in this way. Patients are receiving immunotherapy as a boost, trying to boost the immune system, and then subsequently the patient receives chemotherapy, and thinking that this is going to increase the release of new antigens, immunotherapy is given subsequently, and we are going to recycle. Obviously, this is linked to a lot of translational work with cell-free DNA and also immune parameters in order to know if, in fact, there is some type of increase in the immunotherapeutic effect of avelumab when using chemotherapy given in this sequential way. So the third part is focusing on the new genomic findings, the way to optimize blood cancer patients, mainly the tumor mutational burden, mismatch repair alterations, and DNA repair gene alterations. This is the data that is coming retrospectively from a atezolizumab trial showing that the mutational burden is associated with overall survival. But this is retrospective analysis, and the question is this predictive or is this prognostic? So initially, it was thought to be predictive, so we can see that there is an increase in survival in patients with high mutational burden. But we know that urothelial cancer has a lot of genomic abnormalities. We have the APOBEC signature that is driving the type of mutation seen, and this is linked to an increase in the PD-L1 in patients that have this APOBEC type of signature. And in a retrospective analysis that we published in Annals of Oncology, we saw that mutational burden in patients receiving chemotherapy was associated to better survival. This has been confirmed in the recent TCGA analysis. So the updated TCGA analysis is going to be published in Cell, includes 412 patients. And you can see here the survival. And those are patients that are muscle-invasive bladder cancer patients. They didn't receive immunotherapy. And you can see here that mutational burden per se, or the APOBEC signature, or the MUT-seq clusters, or the neoantigen load is per se prognostic. So this is not a predictive issue. Maybe with immunotherapy, we are increasing this benefit, but this is important to know. We know that in lung cancer, for sure, we have data that mutational load is predictive, but for bladder, you can see here these patients receiving surgery and some of them receiving chemotherapy, the survival is much better if you have more mutations, meaning that your immune system is in better shape if you have more mutations. So what was new at ASCO? Some description that the germline DNA repair alterations are seen frequently, and this is important to remember in order to design subsequent trials. This is another report by Faltas at AACR showing a higher incidence of germline DNA SNPs, repair alterations. Also, mismatch repair is not frequently seen, but occasionally seen in 3%, and this is occasionally linked to upper tract tumors and Lynch syndrome. And if we put all these together, and also this other presentation by Theo, this from the Memorial Group, the DNA repair mutations deleterious DNA repair mutations were seen in 26% of patients with bladder cancer. And we need to take this into the advantage of treating these patients with immunotherapy. And in fact, Ayer, when he presented that, he showed that there was a correlation between response with immunotherapeutic agents and mismatch repair alterations or DNA repair alterations. So this predicts response to immunotherapeutic agents. We know that Pembro is indicated for mismatch repair, so we need to remember that in bladder cancer, there's going to be this 3% of patients having this MSI instability. And why not to combine with PARP inhibitors? So this is one of the arms of this trial that we have seen before. And then the most recent data published by the TCGA is this, this was highly criticized. This is a classification in different molecular subtypes, and I think that we can take the advantage from this TCGA analysis. This was presented by Seth Lerner and asking, is this a future treatment paradigm? Based on RNA subtypes, we are able to further classify on the standard luminal or basal. You can see here luminal papillary, luminal infiltrated, luminal basal squamous, and a new type that has been described, not linked to the morphological aspects of bladder cancer, that is a neuronal. And we can see here, and based on that, and this is a proposal to be explored in future clinical trials. Based on this profile, you can use targeted therapy here. For example, in this case of luminal infiltrated, this is where IOs can work much better. But for example, in the luminal papillary, as mentioned before, the likelihood of responding is pretty low, and probably using agents like FGFR3 inhibitors will be a best way to treat these patients. And this could be applied in the neoadjuvant setting in patients with muscle invasive. So on the way to move forward in the daily basis, what we need to do is to sequence and look for all genomic abnormalities in the tumor, in the germline. And this is a paper that hopefully is going to be published, where we have been meta-analyzing 249 IO-treated patients, with some of them having biopsies before treatment or after treatment at the time of relapse. And you can see here that there are a lot of genomic abnormalities that are linked to a better outcome. We know the mutational burden, but even specific mutations of genes like JAK mutation and so on can predict a response to immunotherapies, and these need to be implemented in all the patients that we are treating. To conclude, this is my last slide. Understanding immunology and genetics has identified groups that respond well to a PD-1, PD-L1 therapy. We have seen that the highly mutated tumors in bladder cancer, mutational burden, MSI, or defects in DNA repair. We have seen this, and this is obviously exploratory, these genetically defined groups described in TCGA that will be published pretty soon. And obviously, we need to implement integrated analysis of response and resistance associated with mutations, mutational signature, clinical annotation, and mutational clonality in all our patients. With that, I conclude my presentation. Thank you very much for your attention.
So with that, I'd like to invite back to the stage for our panel discussion all of our speakers. And in addition to all of our speakers, I'd like to invite also Daniel Chen from Genentech and Nolan Wages from University of Virginia. Hey, how are you? Good to see you. I think are we going to all fit on the second row?
Well, I think we'll all fit.
Can we all fit? Great. One more. 1, 2, 3.
No, let's go to the back row.
So we have some front row, some back row people. So while everyone's sitting, I think what I'll do is I'll summarize a little bit. I heard a lot of common themes in these talks. Some of these common themes were, first of all, patient selection is going to be key for combinations. We already have a response rate that, with duration of response, that the duration is impressive, but with single-agent immunotherapy agents, perhaps the response rate isn't as high as we want. And so we want to make sure we're selecting the appropriate patients for combination therapy or sequential therapy with an eye in mind of who's going to respond best to single monotherapy and who might need combination therapy, and how do we identify those patients from the start. Other common themes I heard were dose optimization is not necessarily just what dose are you giving, but are you giving it sequentially? What kind of schedule are you giving it in? do you need to stop therapy at some point? Because really what a patient would view as clinical benefit is when they're off therapy, and that's something that at the agency we're exploring. What is clinical benefit to a patient, and what could be other novel ways to demonstrate that from a regulatory standpoint? The other common theme I heard was, there are hot tumors that we know will probably respond to immunotherapy agents, but what do we do with these cold tumors, and how do we prime T cells, and what are the different methods we've been using to do that? And then finally, novel trial designs. How do we optimize when we have multiple inputs into a trial? How do we optimize these trial designs so that while we're looking at dosing optimization, how do we take all these factors into account when you have multiple agents, such as 2 chemotherapies or one molecularly target agent, 1 chemotherapy, and an immuno-oncology agent? So these are the themes that I heard throughout all of these talks. One of the questions I wanted to ask has to do with one of the first questions, and this can go to anyone, but in particular to the people who spoke about priming T cells. With this CD40 method to try to overcome low mutational burden diseases, is this priming approach being used in other disease areas?
The CD40?
Yes.
Yes, absolutely. I had the opportunity to tell you about trials that involve pancreas cancer patients, but there's a growing portfolio of clinical trials looking in other tumor types, particularly studies in CD40 antibodies in combination with PD-1 or PD-L1 antibodies.
Okay. And has this been used in breast at all, since you touched on some of the same topics?
There are trials of CD40 in combination with PD-1 and PD-L1, and particularly PD-L1 in a variety of tumor types, including triple-negative breast.
Okay. And by the way, just so that everyone knows, if you have a question, please there's a microphone up there, and my panelists, if you have questions for each other, please indicate if you have a question. The next question I wanted to ask had to do with dose optimization in terms of stopping therapy. And so, Dr. McDermott, have you stopped therapy on patients to see what happens?
Well, I think it's an interesting concept. Looking back to one of the first phase 1 trials with a PD-1 antibody that was done largely at Hopkins. I think there are Hopkins people who can correct me if I'm wrong, but I think on that trial you could get 1 dose of what was MDX 1106 at the time. And if you got some benefit, you could get more therapy. And early in that trial, you saw benefit with just 1 dose. And several people went on to get subsequent treatment. There was a concern about over-treating people at the time. We went from that experience, which was interesting, to the larger Nivo phase 1 trial, which gave up to 2 years. And 2 years, in my mind, was a somewhat arbitrary, end of treatment. Meaning there was no exploration or not enough of a detailed exploration of whether 6 months versus 12 months versus 24 months made a difference. And I think we need to work backwards there, because there are some patients we're clearly over-treating. That said, on that Nivo 003 trial, when patients stopped at 2 years, some of them progressed. So there are probably some patients who are in response at whatever time you want to stop, who need chronic therapy, which is sort of an interesting concept. And in some of those patients, and this was data that Steve Hody presented, I think last year, maybe 2 years ago at AACR, when you reintroduced Nivo in this case, in those patients, some of them regained a response. Many of them did. It wasn't a large number of patients, but that sort of opens the door, in my mind, to looking at different lengths of treatment, considering novel designs like randomized discontinuation designs. Those kind of designs are very hard to execute in humans because they don't like to be taken off treatment when it's working, particularly if it's not adding side effects, which is one of the interesting things about PD-1 is most of the tox happens in the first 6 months. So patients are not as eager to come off, in my experience, PD-1 as they might be Taxol, for example, where they're accumulating side effects. So we need to look at different durations. We need to look at novel designs, because there are probably some patients who need chronic blockade and others that can come off, particularly those with deep responses. Many of those patients, and you learn this from the CTLA-4 PD-1 experience, where there are more deep responses. Some of those patients come off for tox in the first 3 months and never need to go back on treatment. And I think from our point of view, while we need to reduce the tox, that's that's where we need to be going. We need to be going shorter courses of treatment, stopping the drug, see what happens, reintroduce the drug if patients progress. That's what I'd like to see.
Oh, sorry, go ahead. No, go ahead, please.
Maybe I could just add to that. David and I think, have tried to address this question over many years, as have many people in this field. In fact, the original atezolizumab large phase 1 expansion study actually limited therapy to just a year to ask this question. And I would suggest, as David has said, that this is an unanswered question for our field, what optimal duration of therapy is for immune-based therapy. And it probably lies with the idea that in our patients being treated, there are very specific subsets that we don't understand. So perhaps for some of these patients, there really is early eradication of their tumor based on a very intense immune response. And for some of these patients, you see that you can stop treatment early and have very prolonged durability of that efficacy. And that may be because of eradication of tumor. Whereas other patients, perhaps what's happening is you actually have a chronic ongoing battle between tumor growth and tumor efficacy, anti-cancer immune efficacy. And that's just a chronic biologic reaction. And you can imagine that for those patients, stopping therapy could be detrimental. And I think in the larger experience that David is summarizing, some patients, when you stop therapy, have very durable responses. Other patients have some durability, but ultimately progress. Other patients might immediately progress upon stopping the immune-based therapy. And what I think has been difficult is that there haven't been any clear identifiers for which patients belong to which subset. And even the deep depth of response hasn't itself been the perfect measure. So one might assume that a CR might mean eradication. Unfortunately, it might not. It just may mean that the tumor is at a microscopic level that we can't measure. And I think that the clinical experience has suggested all possible outcomes are still in play.
Dr. Emmons?
So we had a really interesting patient on that phase 1A study with triple-negative breast cancer who enrolled on this study when it was written for 1 year of therapy and then stopped. She had pseudoprogression, so she had a nice clinical benefit. If you take the pseudoprogression out of the picture, she had a PR, but was classified as PD because of the pseudoprogression. But she was benefiting clinically from the drug. At about the time she reached her 1 year point, the protocol was modified to allow for continued therapy. So we sort of had the option to decide what to do. And we talked about it, and we decided we might be able to learn more by taking her off therapy and watching her closely. So she came off therapy. We watched her very closely for a year. And about a year off therapy, she developed an enlarged lymph node, which we biopsied, saw tumor cells, and restarted therapy. And she's gone on to have a complete response. So that's one anecdote of a patient that was discontinued from therapy, did pretty well for a pretty good period of time, and then went on to have a better than the initial response when we restarted her immunotherapy after that evidence of progression.
I wanted to make one comment. I find the notion of that potentially we're overdosing PD-1 antibodies. And I think of a paper John Wary and Tara Gangadhar published in Nature just a few weeks ago looking at systemic biomarkers of response in patients with melanoma who were treated with PD-1. And they clearly identified changes in proliferative CD8 cells in the periphery as a biomarker when combined with tumor burden with predicted response. If you look carefully at their data, you'll see that pharmacodynamic effect happens after a single dose. But when same patients were followed receiving the same drug for over the course of several more months, the PD change was lost. So the big bang happens with the first dose in the first 6 weeks, and then it goes flat. And we're not yet sure what that's teaching us, but prolonged use of these drugs, you lose at least that biomarker. I think it's provocative.
So just adding on what Bob said, I think some of this is disease specific. So I think in melanoma, I also treat melanoma. I think there's more evidence that you can stop treatment, particularly in responders, and get a benefit. And more of us, Mike Atkins is an advocate for this, is after about 18 months of treatment in a responding patient, do a PET scan. If the PET scan is negative, consider stopping drug. I think that's the type of thing we need to explore in randomized trials, not just stopping early, but are there other predictors of who will be off? So for example, immunopet isn't something that people are exploring. I know the NCI and other places, that's something that should be considered, but there are a couple of concepts that have leaked into our practice that I think need to be proven correct before we adopt them. One of them is this concept of unlimited treatment. So I mentioned the first Hopkins study, which was 1 or 3 doses or whatever it was. We went to the larger study that was 2 years of treatment. Dan mentioned 1 year. Now we're in a concept where many of our trials are unlimited PD-1. We haven't proven you need that, and I think before we adopt that as a rule, is we need to prove that that's real. The other concept that I worry about is this treatment beyond progression issue, which has been explored in retrospective analyses of giving PD-1 after a patient progresses and showing that the patients who continue on drug do better than those that stop. I think those analyses are very biased. I think they're a setup for people in the community over-treating patients, and I think that's a particular problem because most progression, in my experience, outside of melanoma is real progression, and if you don't move that patient onto something else, they're going to be in deep trouble. And I think this is a problem not just for the patients, but for our field, meaning to make advances going forward, these drugs are so convenient to use. They're getting used so widely in the community. We need to get those patients when they progress for our trials if we're going to make progress. Not when they're treated beyond progression. It's harder to get those patients because I think they fall apart and they can't come to large centers to go on trials. And in a way, by advocating for treatment beyond progression, we're slowing down progress in this field, potentially harming patients. We need to prove in a randomized way that treatment beyond progression is useful before we adopt it as standard of care.
Dr. Jaffe?
Yes, I'd like to raise a slightly different way at looking at this problem. Is this a problem of over-treating, or is it a problem of not adding the right combination drug? Because the emerging data suggests that PD-1 blockade isn't able to keep cells going, stay activated. And maybe by repetitive treatment, we're trying to reactivate or bring in new activated cells. And in fact, I think the data is emerging from John Weary, from Rafi Ahmed, and also preclinical data that PD-1 blockade can push these T cells into exhaustion. So maybe what we really need to do is limit that therapy once it reaches a certain response rate where we can't move it anymore, but think about how do you make sure those T cells don't become exhausted. And obviously OX40 agonists, there are other agents as well, but I think we need to really think a little bit differently about this problem and say, yes, we don't want to continually treat patients if we don't see benefit. Biomarkers, of course, would be the best way of knowing if this is happening, and we need better biomarkers, and I think there are many people looking at this as well. But I think we have to pay a little bit more attention to what is happening and not make decisions based just on clinical outcomes.
So speaking about biomarkers, I mean, that's a theme that I think we're going to be hearing a lot about today. And so we need better biomarkers to identify which patients to treat either with monotherapy or perhaps with our current standards of VEGF TKIs, let's say, or which patients do we need to select for combination immunotherapy, but also clearly which biomarkers can we use to select length of treatment, when to add something, and those things. So my question regarding this is, do we continue to embark on trials using PD-L1 as the marker, or what other markers have you been using to try to identify those patients from the start who should be not just, I mean, we know mutational load, and we'll probably hear a little bit more from Dr. Chen about things that they're using in his company to identify the proper patients. But what other biomarkers are you exploring in the lab right now to identify the appropriate patients for combination therapy?
PD-L1 is the first biomarker, so folks are still looking at that. Whether or not there's T cells present there is an important consideration, and what the phenotype of those T cells is in terms of what checkpoint molecules they express is also pretty important, like what I showed with the preclinical data, combining sting intratumorally with PD-L1 and OX40. So I think you can map the immune microenvironment of a tumor and design things based on that, ideally.
So can you envision a trial, sort of the multifactorial trial that you discussed, in which you would use these different biomarkers with different doses to select an appropriate dose for different populations, or do you think it has more to do with the biomarker in the patient and whether or not the therapy is appropriate for them at all?
I think that kind of unusual trial design, which you could try to optimize a combination preclinically using that approach, and then maybe in a simplified approach in the clinic, I think I would use biomarkers for patient selection and then explore a directed combination based on the biomarkers that we targeted for selection for the combination.
Okay. All right. My next question had to do with you brought up a little bit about other extrinsic factors or intrinsic to the patient, such as obesity and gut microbiome. Has this played out at all in breast cancer to date?
So in breast cancer, that's definitely an emerging area. Breast cancer is lagging a little bit behind in terms of clinical responses with these agents. So there are a lot of people that are interested in looking at that and incorporating that into the trials, but there's not a lot of data about it so far.
And for those of you in other disease areas, has that come into play at all?
We are, in fact, retrospectively analyzed patients that did receive antibiotics before starting IOs, and because this might impact in the microbiome, right? So and I think there is in other diseases that there is an impact. If you give antibiotics, you might change them, the flora, intestinal flora, and this might lead to a different outcome. So there is an emerging issue. Now we have a trial that we are prospectively doing, taking the rectal flora in these patients and try to see the B cell population there that is impacting on the outcome. So yeah, it's absolutely an emerging area that we need to explore more, because, or even low-dose steroids, what's the impact of low-dose steroids, antibiotics, even, let's say, even 3 weeks before starting therapy or during therapy. So an area that need to be explored further.
Dr. Xu?
Okay, my name is James Xu. I'm one of the reviewers from FDA. So I have a question and comments for David. So you mentioned about the we need to select patients between VEGF inhibitors and PD-L1 immunotherapies. So this is very remarkable and insightful. So I was the reviewer for nivolumab in renal cell carcinoma in the second-line setting. So back in November of 2015, when FDA first approved this drug, we actually made the exploratory analysis. So our hypothesis was that based on patients' prognostic risk categories, you can actually select between VEGF inhibitors versus PD-L1s. We made this hypothesis based on the PFS analysis. So what we noticed is that for patients who has a poor risk categories, that the nivolumab was doing much better in the PFS versus the everolimus. So what is your comments on if you think it would be worthwhile to study maybe a sequential therapy comparisons using second PFS as an endpoint? We actually published this hypothesis in our in the Oncology Journal this year. So just want to get your comments on that.
I mean, I think sequential trials are important, not just in kidney cancer, but in a variety of tumor types. I think they're hard to execute, though. We've certainly proven that several times in the kidney cancer realm, where you take approved drugs and you ask patients to consider a sequence. We've done TKI versus mTOR with a switch, for example, at progression. And what happens in most of those trials, particularly when you're dealing with approved drugs, is patients don't comply with the switch. They often drop off. And that confounds the results of the trial. Those numbers improve when you're dealing with unapproved drugs and patients are motivated to switch. So in the case of the Emotion 150 trial that I mentioned, there's actually a crossover in that study that's producing some interesting insights about resistance, not just to VEGF, but to PD-L1. patients were motivated to switch, and 70% of them did because they could get the novel combination that they couldn't get otherwise. But I think if we're dealing with a trial, for example, let's say Sutent, then Nivo versus Nivo, then Sutent, that's an important trial to do. I just think in reality it's going to be hard to execute if those drugs are available outside of the trial, because patients will drop out. They'll choose to do what they think is right, even if. So it's a hard question to answer. But it gets back to the larger point, which is, in kidney cancer, we're likely going to end up in a world a few years from now where the FDA is going to approve 4 or 5 different combos of VEGF plus PD-1 in the first line. In my mind, if one of them gets approved, they're all going to get approved. So assuming they're all approved, there are going to be patients who are not going to be benefiting from either approach of those combos, but they're going to be getting them anyway. So there will be patients who are resistant to VEGF strategies, but they're going to be getting one because everyone's going to be switching to combos. There are going to be patients who are PD-1 resistant, which over a third of kidney cancer patients are, that are going to be getting PD-1 in the front line. And in the end, I don't think we've done them a service if we're doing that, not just in kidney cancer, but in the larger field, because the sequence might be just as good in many of those patients.
So what about if you allow for a crossover and using the second PFS as an endpoint?
Well, I think like I said, I think in theory that's a good trial to do, and if you could execute it. So that ultimately you'd want to do a trial that compared, say, exitinib pembro versus exitinib and then pembro, or pembro and then exitinib. That would be a very useful trial to do. I just think it's hard to get patients to sign up for a trial like that.
Thanks.
But this overall question really highlights, I think, a very interesting piece for this entire field, which is that our patient populations are changing. And so this question around prognostic variables, essentially as I understand it, a second-line renal cell setting where they got a VEGF TKI in the front-line setting, could be something that predicts for cancer immunotherapy, but it also could predict for what your outcome was from your frontline TKI. And so, David, do you have a comment on whether, if by looking at prognostic variables in the second-line setting, could that actually just be reading out whether Sutent was an effective drug in the frontline setting versus less effective?
It's possible. I mean, I think, as James mentioned, one of the interesting parts about the Nivo phase 3 in kidney cancer was that the greatest activity of the drug was seen in our worst patients, or by clinical criteria, patients who you expect to do poorly. And that's a very exciting aspect of this whole PD-1 story, is that patients whose tumors would behave poorly with standard therapies are some of our best responders. That's truly exciting. In some tumors, that may connect to this whole mutation burden story. That's probably not the case in kidney cancer, where kidney cancer is not a very mutated tumor. It may be a more specific mutation story in kidney cancer that may play a role, and there are people looking at that. As far as prior treatment, I think if I understand your question, Dan, I'm not sure I did, but there's certainly BMS has looked at the impact of prior treatment on the response to PD-1 blockade and seen some interesting findings where patients, for example, who got pazopanib before PD-1 seemed to do better when they got Nivo than those who got sunitinib, and then PD-1. Whether that's real or not, I don't know, but it generates an interesting hypothesis about whether pre-treatment makes a difference in response to PD-1, which gets to what Leisha and Bob were talking about as far as priming a tumor to respond. Is there something about prior treatment that might set you up to be a better responder? And I think that's an area we need to push on, because most of our tumors are cold, most of our tumors are non-responders. Even those that are responding will eventually progress. So we need to look at those clinical criteria, including prior treatment, to see if it matters, because it may lead us to better combinations, better sequences in the future.
So I had a question for all of you, and this came up in several of your talks, and it's come up inside the agency certainly a great deal, is our greatest resource is our patients, and with the large number of possible combinations that can be studied in each of your disease areas, how are you prioritizing which trials, either at your institute or you personally are putting patients on? How are you prioritizing which trials you'd like to go forward with for these combinations?
So I can speak for myself. So I'm trying to prioritize trial selection based on the scientific rationale. So, and that evolves with the data to some degree. So if I am looking at a combination trial, rather than just combining something because you can, I look for a good scientific rationale for why there could be potential immunologic synergy behind the combination. So that's generally how I'm choosing. And then, in breast cancer, we have the advantage of classifying these patients, although it's not a perfect classification, and that helps guide which direction to move the patients into. So for HER2-directed therapies, we use HER2-based combinations. Triple-negative breast cancer is kind of wide open, so there's not quite so much there. That's purely immunologic mechanism-based. And then ER-positive breast cancer is getting more complicated because now we have not only hormonal therapy, but we've got CDK4/6 inhibitors. So I look for trials that fit into the treatment paradigm, but also have a good immunologic rationale for potential synergy.
There you go. I was going to add one thought. So those are the types of considerations if the denominator stays the same. So, the question is, do we have more trials and more combinations than patients who can go on clinical trials? But in this country, the percentage of patients who go on clinical trials is 4% for cancer. 4. So we're not testing new ideas in 96% of our patients. Now, at high centers of excellence, some of the ones represented here, that number's a lot higher. But if we could change the denominator and understand why that number is 4%, there would be many more patients interested if we could solve some of the problems which go beyond the intricacies of trial design and get into issues of access and availability and geographic challenges and inclusion-exclusion criteria, which may be limiting us in ways that now in this field we could begin to open up. So I would put that out there as another way to think of a solution.
So, I completely agree with what Bob said, and I think this gets to what I was saying before about this concept of, how easy these drugs are to give in the community and patients being treated beyond progression, not being referred when they should be. I think we're sort of a victim of our own success in the sense that when these trials were phase 1, we had more patients than we had slots. Now we sort of have the opposite problem, and I think we can't, as a field, contribute to that problem by encouraging things that are unproven. But to get to your question about how do I decide about whether I want to explore a specific combination, I think most of these combinations that are out there in development have very similar characteristics, so it's not easy. They all have interesting preclinical data and multiple tumor models showing that they can make the tumor go away, which is great. Many of them have single agent toxicity profiles that are manageable. Most of them don't have single agent activity. So if you ask, all right, so which ones will you want to study? To me, the ones I'm most interested in are combos that have activity in PD-1 failures. I think those are important. It's an early signal of activity of the combination. I think looking at combinations that have activity in biomarker-negative patients. I know our biomarkers are not great, but they do enrich for responders. So if you can have responses in PD-L1 negative patients, I think that's meaningful early on. Because I think, and going forward, ideally, the sponsor of the trial would have a sense of who's going to benefit. So they'd have a sense of a marker ahead of time on who they want to treat, not just treating everyone. Because I think trials, we've had great success in this field with lots of pivotal trials in unselected patients in the salvage setting, many positive trials. Almost every single trial has been positive in that setting. But as we move to frontline, as we move to adjuvant, if we move without an ability to select, we're going to see the same percentage of trials be negative, in my opinion, as we are now seeing positive trials, because these combos are not active enough to win in all comers, I don't think.
Yeah, let me add something else here. In addition to these trials that are genomically driven with some potential markers, I mentioned this SPY trial in breast that could be applicable in bladder. There is another thing that is the concept of sequencing. One of the questions that has called me a lot of attention is you see that at least in bladder and also in lung cancer, we can see that in the trials where we are comparing immunotherapy with chemotherapy, in the very beginning, the survival curves is worse for the immunotherapy. So there might be a need to prime the tumors with chemotherapy before moving to immunotherapy. But in these first 2 months, that is the time that you require to boost the immune system. Usually in lung cancer, in bladder cancer, the survival is worse in patients receiving immunotherapy. So something is going on there that probably if you, and this type of concepts, and I mentioned this other trial design, like trying to use the chemotherapy to prime the immune response subsequently, is something that we need to think about. It's not just combine two drugs, give both at the same time. Sequences, I think those are very, very relevant in clinical trials.
So how easy? So we've heard from Dr. McDermott that getting sequencing trials and/or randomized discontinuation trials are difficult to do. Who's sponsoring the sequencing trials? Is it cooperative groups? Is it industry? Is it investigator sponsored?
So this one that we are doing is an IST. We approach a company and say, okay, we have we don't want to combine immunotherapy with chemotherapy, and that's it. Let's try to, in addition to the combining. Look if there is this trial where we are going to give immunotherapy before starting chemotherapy, and then on day 15 we are going to give again immunotherapy, looking for like a increase in antigen release by the chemotherapy before giving the immunotherapy. So yeah, so this type, and obviously you need to have strong monitoring of how the immune system is changing with all these sequences and so on. But this is an IST trial, so it's, I don't know, companies maybe are not very, very interested with that.
I just wanted to return to one point that you made, Dr. van der Heide, about the recent study where looking at PD-1, after just the first dose, you may have gotten most of your impact. So do you think there will ever be, we've discussed length of treatment, do you think that, treating as much as one dose or 3 doses or, 6 months will be within a randomized discontinuation trial?
Well, yes, I hope so. I think we have to refine those biomarkers. That was a blood test. Those investigators are now looking at tissue-based, see if maybe something deeper was going on in the tissue. But yes, what we're understanding, and CD40 is a good example. Everybody immunologically responds to anti-CD40. I think it's 80% of patients have that response to pembrolizumab, but not nearly as many people respond. As we refine those biomarkers, particularly early looks soon after treatment, I think there is a way to go forward using ever more sophisticated biomarkers to design trials around discontinuation. And as Liz said, move on to the next step. We got what we got out of PD-1. What's behind it now? Is it CTLA-4? Is it VISTA? Is it, what's next? And begin to drive trial decisions based on those biomarkers. I think it's a very real possibility.
Just to go back to the denominator issue that Bob raised, if you see at least in my experience, if you look at the referrals coming in and talking with my colleagues in the community, there are a lot of patients out there who don't get the best access. And in fact, they either get the drug off-label, in which case we're not really learning anything from that, or they get referred very late when really they're not very likely to respond, their performance status isn't very good. So I think the more that we can create relationships with the community oncologists and really work together to pull patients into the clinical trial system, that'll help advance the field and kind of at least start to equalize the abundance of resources with the drugs that we have to test with the patients that we have to test them in.
And to your point also about eligibility criteria, we've been working on a project of sort of modernizing eligibility criteria such that to encourage both companies and investigators to think more carefully and scientifically about which eligibility criteria as you move through development are most important to encourage this to opening clinical trials to more patients, including, brain metastases and HIV and prior malignancies and organ dysfunction, and what's most appropriate for that agent, and to think more carefully so that, and age as well for appropriate diseases, to think more carefully about which patients really should be included in trials, and so that potentially we would also have more representative data pre-market before a product gets approved and goes to the entire market, where patients who may not have been represented on the trials will be treated. And especially with immune agents, we have there are very few patients who had autoimmune diseases who were treated in clinical trials, and we know that it's being used out in the real world with patients who have autoimmune diseases. So these kind of issues are things that we're trying to address at the agency as well. Okay, if there are no other questions or comments, are there any other questions or comments?
Can I ask a question of the front
please.
row?
Yes, please.
As we begin to think about checkpoint blockade combinations with chemotherapy, one thing that I wonder, medical oncologists will often use steroids for antiemetics in not trivial doses. Some of these drugs are weekly, and so I'm looking at Leisha and Joaquin. What do you think? Are we ignoring that? And is that an impact? I worry that it's an elephant in the room we haven't really got our head around yet.
So the data on that are a little bit mixed. There's preclinical data that you can give immunotherapy and a significant dose of steroids, and it doesn't impact the induction of immunity. A lot of the early data has tried to use agents that don't require steroids, like the combination with nab-paclitaxel. That has the immune modulatory benefits of the taxane, but you don't have to give steroid pretreatment. With paclitaxel, a lot of time we can wean the steroids off, although we have to give them in the beginning. There are clinical trials that are looking specifically at the issue of steroids and what their impact is on the response to the agent. So, a PD-1 or PD-L1 in combination with a Abraxane, nab-paclitaxel with steroids. So that should give us some information. I think it's being systematically looked at in least one trial that I've seen.
Yeah, so we try to avoid giving taxanes in bladder, so unfortunately we don't use it that frequently because giving a bolus of 20 milligrams of dexamethasone or 10 milligrams, for me it doesn't make sense. There is data in melanoma saying that you can give up to 10 milligrams of prednisone and it is not impacting on the outcome. And this is the only data that we have. Other is speculative, and I try to avoid as much as possible to give steroids, but I understand in breast cancer that you give a weekly Taxol, at least you need to give 8 milligrams or whatever. So this might impact on the outcome, but we don't have data on there.
It's still an open question.
Chirag Patel, Takeda. This is a question with regards to clinical trial design. As we've heard that it's so important to select the right patients for your clinical trials, and also the importance of combining or sequencing these different agents. I'd like to know from the panelists what your thoughts are, especially with regards to prioritizing these kinds of questions when you design your phase 1 trial in the process of optimizing the dose.
I'm going to take this off.
See if I can get my, is this working? There you go. I'm, of course, coming at this from a biostatistics point of view. So I think it's a good question. As we've seen, the trials are getting more complex, and there's a need for novel designs. I think I might even pose a follow-up question to the group about the role of exploring more combinations in this setting, because we saw in a couple of the talks very small numbers of combinations being explored with a little bit of safety data being accumulated at the beginning. Like in the first talk, there was a two-arm safety study with 10 patients in each study. I saw 2 or 3 dose levels in combinations in later talks that had 3+3 designs. And then I was struck by the contrast between those and Dr. Emmons' talk that explored the combination grid of 3 dose levels of 2 agents, and she made a great point about how one of those dimensions could be schedule or something else. But when we're asking these more complex questions, I'm wondering what the role of exploring more combinations is, and of course the trial design needs to catch up with those more complex questions. I think it's lagging behind a little bit at this point, and so I'm wondering if the right questions aren't being asked, or are we sort of boxing the question into the trial design that we know how to do, and so I think that's an unanswered question from my standpoint, and one that is very important.
So one of the advantages of that factorial design that I showed, and if it can be applied in the clinic in an effective way that allows you to get information quickly, and I think we're better positioned now to do that than we were when I did it, is you can bracket what you think is likely to be the bioactive dose, and then when you generate the response surface, it actually is across that entire dose range. So it could end up being that the best dose combination is actually not one that was tested. So you can identify that, and then you can take it forward in a small cohort of patients and get more information about it. So you could almost use that as a really exploratory way to look at the interactions between dose ranges, identify what looks like the best, test it in a small cohort, and if it looks good, then you can take it further. So it's one way to potentially more quickly look at different doses rather than the traditional stepwise 3+3 or the other different types of dose escalation, de-escalation that you see.
So, yeah, go ahead.
I was just going to follow up. How many patients were on that factorial design study?
28 patients, 22 were actually in the grid. 6 people got the vaccine by itself, so they weren't included in this surface.
Oh, okay.
So it was 22 patients. And, you know, we included 2 or 3 patients at each of the points, except for one, which only had 1 point. But every single point informs the response surface, so it actually has a lot of power to find good data.
Right.
So I just wanted to add that I think that overall this meeting is tackling probably the most difficult problem for cancer immunotherapy. And it's based on a number of things. First, we got to talk about single agent activity. We're talking about combinations. You're talking about a class of drugs that have acute effects, both on efficacy and safety, and chronic effects on efficacy and safety. And when we think about the chronic effects on efficacy and safety, some of those are going to be dependent on continued dosing, and some aren't. So you can quickly see just how many variables there are going to be. So just some perspective on how we as a field can start to approach this. When you look at the possible different combinations, when you add dose and schedule, the number is astronomical. And so going into these experiments, these clinical experiments, it becomes even more important, number one, to start with a hypothesis. Number two, based on that hypothesis, have some near-term biomarkers so you can measure that effect in humans in a small number of patients where you can actually vary some of those parameters in an underpowered fashion. And then 3, look for big effects, right? In small numbers of patients, you really can't tease apart more nuanced effects, but you can tease apart big effects. So dose and schedule where one dose and schedule works and the other dose and schedule doesn't work. Those are the kind of things that we should be able to tease apart. But be very cognizant that those acute endpoints, and we've talked a lot about inferences made upon acute endpoints, and we've already said response rate's probably not the most sensitive or specific biomarker for efficacy in cancer immunotherapy. But then we go on to talk about all these inferences around biology related to response rate. And so be very cognizant that, of course, the real biologic endpoint for us in these clinical trials is OS. And even with OS, that can be split into OS tail of the curve, OS hazard ratio, and those are 2 very different things in terms of the long-term impact to the patients that we're trying to help. So this is a very complicated topic, and you know, we can hopefully get to a point where at least we have a framework to think about it, but it's unlikely that today in 2017 we'll have the best answers on how to address all these problems.
Thank you. Okay, we are over time. If you have a super fast question, or is this a long and complicated question?
I'll make it very brief. Amy Van Andel, Boehringer Ingelheim Regulatory Affairs, and I just wanted to provide a quick comment from the industry perspective about the access to clinical trials. And I know you talked about inclusion-exclusion criteria, and it goes along with all the hypotheses have been generated and the great desire to have public-private partnerships, et cetera, to do these studies and the community involvement. And I just want us also look at the operational aspects of doing these things and find innovative, efficient, non-bureaucratic ways to be able to access these patients and modernize the way we conduct trials and use technology, virtual aspects, et cetera, so that we can lower the burden to being able to have these patients in other settings being able to engage in our trials, either in the US or globally. And I think that's an area where we really have great gains to be able to make, and it will require all of us, industry and community, regulatory and sponsors, to work together to think of ways that we can conduct these trials just as clearly, with just as high quality, but lower the barrier to participation in them. Thank you.
Okay, so I think I'll sum up. It's complicated. Thank you. Thank you for making that point. This is complicated. Combination therapy in immuno-oncology is complicated. And clearly we're all trying to deal with this. I think the take-home that I have from this session is basic science is really going to drive how to use these combinations, and we need to continue our efforts with many of the people who are here today to figure out which combination works for which patient population in which disease. And so I'll leave with that. Thank you very much to all of our panelists. I appreciate your time. Thank you. So great. So we are supposed to be reconvening at 10:30, and since we're only a few minutes over time, I think we'll still reconvene at 10:30 for panel 2. Thank you.